What is the neuromuscular junction? Well, in order for these 4 individuals to do this, and these 2 individuals to be able to do this,
23. FRN has recovered over 6,000 pounds.
a whole lot of things are going on molecularly to make that possible. We are going to dive into what exactly makes these things possible. So to start out with, we have an action potential that comes down the neuron that is connected to the sarcomere. And this action potential opens up voltage-gated calcium channels on the axon terminals. Now, a voltage-gated channel is going to open whenever there is a change in membrane potential, which is another way of saying an action potential is there, and that's what's causing a voltage-gated channel to open. In this case, it is a voltage-gated calcium channel that's going to open up on the axon terminal, and it's going to cause all of these calcium ions to come into a presynaptic terminal. So all these calcium ions come on in, and these calcium ions are going to bond with vesicles. Now, inside of these vesicles contains the neurotransmitter called acetylcholine. Now, acetylcholine, once the calcium binds with the vesicles and interacts with them, the vesicles are going to fuse to the presynaptic membrane on the axon terminal, and the vesicles are going to release neurotransmitter acetylcholine via exocytosis into the synaptic cleft. Once all the acetylcholine makes its way into the synaptic cleft and comes out of the vesicles via exocytosis, the acetylcholine is going to bind to ligand-gated sodium-potassium channels. Now, a ligand-gated channel in general is a channel that is going to open when a chemical or a neurotransmitter binds to it. In this case, we have acetylcholine that is binding to the ligand-gated sodium-potassium channel, causing it to open up. Before we talk about sodium and potassium ions moving through these channels, we first have to make sure that we understand what a sodium-potassium pump is. Now, the way that the sodium-potassium pump works, to put it briefly, is that it establishes resting membrane potential. The way that the sodium-potassium works is that it moves 3 sodiums into the extracellular space, and it moves 2 potassiums into the intracellular space. This creates the resting membrane potential that we could see here on this graph, which is usually around negative 70 millivolts. So if we understand how the sodium-potassium pump works and how it establishes resting membrane potential, that is going to make the process of these ion movements make more sense. So before any ions move, the outside of the muscle cell is more positively charged, and the inside of the muscle cell is more negatively charged because of the work by the sodium-potassium pump that establish resting membrane potential. Now, once acetylcholine binds to these ligand-gated channels and they open up, sodium is going to influx the cell. It's going to influx, and it's going to come in, and it's going to keep coming in, and it's going to keep coming in. And guess what? It's going to keep coming in. At the same time, potassium ions are going to leave the cell and go into the extracellular space. However, the important thing to note is that sodium influx is going to be greater than potassium efflux. That is what creates the graded potential, which is called the end plate potential in this graph. The graded potential, or the end plate potential, is built up by sodium influx at the motor end plate via the ligand-gated sodium-potassium channels. It's getting positive because more positive ions are going inside of the cell, which is depolarizing it and building it up to where threshold is. The buildup to threshold is called the end plate potential. So this process is going to keep happening. Sodium's going to keep coming in, Potassium will keep leaving. And then once the membrane has depolarized enough to where we hit threshold, that is when a wave of depolarization is going to occur across the sarcolemma, and it is going to open up voltage-gated sodium channels. The opening of the voltage-gated sodium channels happens because we've hit threshold, which releases an action potential. The opening of these channels allows for further sodium depolarization of the sarcolemma, further depolarizing, getting more and more positive. This is happening across all voltage-gated sodium channels, which are these purple channels in this picture, in this video. And as that's happening, we are in this phase of the graph where threshold has been reached, voltage-gated sodium channels have been opened on the sarcolemma, allowing for further depolarization and sodium influx. Once we've hit threshold, an enzyme called acetylcholinesterase is going to come in and kill all of these acetylcholines in the synaptic cleft and remove the acetylcholines that are opening the ligand-gated channels on the motor end plate. So as further depolarization is occurring because of the sodium influx, voltage-gated potassium channels are also going to open, and potassium is going to efflux from the sarcolemma, and it's going to go out. And this process is going to repolarize the membrane, and we're going to get more negative again. Eventually, our sodium-potassium pumps will reestablish resting membrane potential. So when threshold is hit, the action potential is going to be sent all throughout the sarcolemma, and it's going to travel down, and it's going to hit what is called the T-tubule. And the T-tubule is an invagination of the sarcolemma, where it allows for action potentials to go deep into the muscle cell and hit what is called the sarcoplasmic reticulum. The sarcoplasmic reticulum is an extension of smooth endoplasmic reticulum in muscle cells. It is a special kind of endoplasmic reticulum because it stores calcium ions inside of it. Once the action potential hits these sarcoplasmic reticulum, do the same thing on the other side. It's going to travel down, go down the T-tubule, hit the SR. Once that happens, calcium is going to leave the sarcoplasmic reticulum, and it's going to come over, and before it binds, we got to understand the anatomy of actin. So before any calcium binds to anything, we got to understand that actin molecules have these things called active sites, and active sites before calcium bondage are covered by the protein tropomyosin. The tropomyosin prevents myosin heads from attaching to actin and prevents anything from contracting. We have our calcium ions that are going to bind to troponin. And then once calcium binds to troponin, it is now going to look like this, where the tropomyosin has slid off of the active sites on actin. The active sites are free for myosin heads to bind to them. Once we have this setup, we can now go into excitation contraction coupling. With excitation contraction coupling, we start out with cross bridge formation. We have an energized myosin head with ADP and phosphate on it. and the myosin head is binded to the active sites on actin. Then for the next stage is called the power stroke. And for that power stroke to occur, the ADP and the phosphate release from the myosin head. When you slingshot a rubber band and you let go, it releases energy and the rubber band goes forward. That is what is going on with this. ADP and phosphate, once released, pulls the myosin head forward. It does its power stroke and it pulls the actin closer to the M line on a sarcomere. The next stage, and in order for the myosin head to detach from the actin, we need ATP. ATP is going to come in, it's going to attach the myosin head, forming a cross-bridge detachment. And then ATP is going to hydrolyze and is going to recock the myosin head back to its original position. ATP hydrolysis just means you put a water molecule on this ATP and you split it into ADP and phosphate. That process re-energizes the myosin head to then be able to attach to actin. And then the process can repeat itself again. So that is the neuromuscular junction and excitation contraction coupling.